everybody. I looked at your feedback. Thank you for answering. Most of you said that you have a great understanding of this unit so far, which is amazing. And some of you had a couple of questions. So in this video, I just want to review everything that we learned. I'm so happy to hear that most of you are really enjoying this unit. I know that a lot of you remember some of this stuff from middle school, which is great. I also know that you guys want to get more into detail and go into some cool stuff, and we will. We just have to work our way up there. You know, this week was just kind of an introduction, and then next week we'll start getting into more complicated material. So I got a couple of questions about what histones are and how they are involved in gene expression or gene regulation. Histones are proteins, okay? A protein that provides structural support to a chromosome. So your DNA, right? We said that usually we don't see DNA in the form of a chromosome. We see it in the form of chromatin. So the histone is like a spool, and the DNA is like thread. It wraps around the histone. It wraps around the spool. So the histone is a protein, okay, that's spool. And then the DNA is the thread. It's wrapping around the spool. Basically, think about it. Think about how much DNA we have. Also, think about the storage. We're storing all this DNA inside of a nucleus, and that's super tiny, and we have so much DNA. So in order for the DNA to fit inside of the nucleus, it needs to wrap around these histones to give the chromatin or chromosome a more compact shape. Here, right, the chromosome is the super condensed version. This is what it looks like when DNA is replicating. But on a normal basis, it's in the form of chromatin. And that's the same thing. It's the DNA wrapped around the histone, the protein. So how does this come into gene expression? Well, if you look at this picture, right, there's these little histone tails, these little strands that hang off of the histone. In this picture, you could see how tightly wound the DNA is around the histones. The histones are pulled super close together because the DNA is wrapped tightly around it. And the DNA becomes inaccessible, and the genes will be inactive because they're tightly wound. It's hard for the DNA to be expressed. It needs to be loosely wrapped around the histone. So here, or you'll have an acetyl group, a methyl group. These will attach to the histone tails, which will cause the DNA to spread out and not be so condensedly wrapped around the histone that the gene will be able to be expressed because it's not being blocked by anything. Like here, it was being blocked because the DNA was wrapped around the histone so tightly that they were close together. The DNA wasn't accessible. So I know that was a little bit about last unit, but I just wanted to address that for those of you that had questions. Now, obviously, this is a really complicated process. This is simplified. Epigenetic changes, right? Changes in how if a gene's being expressed or not, can just be caused by development, how you're brought up, the environment, drugs, aging, your diet. All these things play a role in what genes are being expressed or turned on and turned off. Now, changes in epigenetics can result in autoimmune disease, mental disorders, diabetes, cancer, depending on, what genes are being turned on, what genes are being turned off. And somebody also asked a question about what if, this is going to what we've been talking about, but what if somebody has two different color eyes, right? What if somebody has one blue eye and one brown eye? How does that work? This actually is an example of epigenetics, where a gene, due to whatever reason, was turned on. It also could be from diseases, but a gene was turned on instead of being turned off, and it could result in having two different color eyes. Portions of DNA are genes, but a section of DNA is called a gene, okay? Genes code for proteins, okay? They also determine your traits. So what are examples of genes? Eye color, hair color, height. Those are all different types of genes, different portions of DNA that are determining that trait, determining how tall you are, your eye color. Now, alleles are a form of that gene. A gene would be eye color, and allele is a specific form of that gene. So that would be blue eyes, brown eyes. These are alleles. Now, where do these alleles come from? Well, going back to meiosis, in our body cells, we have 46 individual chromosomes. But remember, meiosis produces egg cells and sperm cells, which each have 23 chromosomes. When these two sex cells combine, well, that creates a zygote, a baby that's going to have 46 individual chromosomes. Now, remember when we talked about those genetic laws in meiosis that we will talk about again, but one that I want to bring up is law of segregation. Remember stating that two alleles need to separate during meiosis to end up in different gametes. So allele, specific form of a gene, right? 46 chromosomes. You have 23 pairs in your body cells because you get one from mom, one from dad. You can't get two different eye colors from mom and then two different alleles for eye color from dad, because in meiosis, you're only getting one chromosome. And then those come together, one with the genetic information from mom, one with the genetic information from dad. So based on, crossing over, independent assortment leads to genetic variety. You end up with one in your egg cell or in your sperm cell. You end up with one number one chromosome, 1 through 23, right? And on that chromosome, it'll only have one allele for eye color, one allele for height. So that way when they combine, you get two alleles for eye color, two alleles for height, one that came from mom, one that came from dad. Okay, now genes are responsible for the expression of traits. Alleles are responsible for the variation in which that given trait is expressed. Two chromosomes, let's say this is the number one chromosome from mom, and this is the number one chromosome from dad. Alleles are located in the same location on a chromosome, which is something that we talked about, right? These are homologous chromosomes. They code for the same genes, which means that their alleles are going to be in the same position. So this would be mom's allele for eye color. This would be dad's allele for eye color. They're located in the same location. Now, genotype versus phenotype. Because of the different effects of dominant and recessive alleles on an organism's traits, an organism's traits do not always reveal its genetic composition. So that's why we need to distinguish between an organism's phenotype and an organism's genotype. Phenotype, and I said this yesterday, phenotype equals physical. That's going to be your physical appearance, the physical trait that you have. I have brown eyes. That's my phenotype. But the genotype is the genetic makeup. The genotype is going to be the alleles that you have. So I have brown eyes. That doesn't mean that I have a homozygous dominant genotype. It doesn't mean that. It could mean that. Or my genotype could be capital B, lowercase b. It could be heterozygous. So the genotype is the genetic makeup. It's the alleles that you have, the one that you got from mom and the one that you got from dad. So my dad has blue eyes. So I know because my dad has blue eyes and my mom has brown eyes, that this is my genotype. And here is an example. 2 capital P's for a pea plant flower stands for purple. So PP is the genotype, and the phenotype, what characteristic physical appearance it has, it appears purple when it has the PP genotype, capital P, capital P. But if a plant has capital P, lowercase p, dominant for the capital P, and then a recessive, which can stand for, actually in this case it's white. So lowercase p is recessive, it would be white. My flower is not going to be white, it's going to still be purple. But these are two different genotypes. One's homozygous, one's heterozygous. So if the two alleles at a location differ, if you have mom that has brown eyes and my dad that has blue eyes, one's going to determine the organism's appearance. That would be the dominant allele. Brown eyes is dominant to blue eyes. So the dominant allele is going to be the one that appears. The one that has no noticeable effect on your appearance, on your phenotype, is your recessive allele. Your phenotype is going to be, your physical appearance is going to be based on the allele that is dominant. The recessive one has no notable effect on the appearance. Some of you are wondering about the example that it gave in the video with people who have 6 fingers. So having 6 fingers is a dominant allele, meaning that if you have an allele for 6 fingers, that's going to be dominant. The recessive allele would be 5 fingers. we don't really see a lot of people with 6 fingers, even though it's a dominant allele. Well, that's because it's not very common. What that part of the video is trying to say is that people often come to the conclusion that a dominant trait also means that it's going to be common. But in this case, the dominant trait is not common. Whether or not a trait is common has to do with the amount of copies of that allele that are in the population. There's not a lot of copies of the allele for 6 fingers in the population. It has nothing to do with whether the trait is dominant or recessive. It just has to do with the amount of copies that are in the population. And if you're still interested in learning more about that, you can click the link up here. So monohybrid crosses. This is testing one gene. Okay, so in this example, it's going to be the gene that's the color of a pea plant. So the first thing that you would do is make your Punnett square, and you would put the parent genotypes, let's say this is mom and this is dad, on one side. Both letters for mom on one side, and then the both letters for dad on the other side. So green peas are dominant to yellow peas. Okay, so mom is a green pea, and she is homozygous dominant. So what true breeding is, see true breeding green pod, that just means homozygous, that it's a true breed. It's not a mixed breed, right? If something's a true breed, it's going to have 2 of the same alleles. Dad over here is homozygous recessive. He has 2 recessive alleles and his phenotype is yellow. So we create this Punnett square, this monohybrid Punnett square, to determine the probability of these pea plants mating, creating a new generation, what the probability of each genotype, the genotype that the offspring will have, and the probability of the phenotype that the offspring will have. Capital G, lowercase g, capital G, lowercase g, capital G, lowercase g, capital G, lowercase g. In this instance, they all have the exact same genotype, capital G, lowercase g. What is the probability that the offspring will have the genotype, capital G, lowercase g? Well, there's 4 boxes. Each box is worth 25%. It is 100% probability that their offspring will have this genotype. What phenotype is the offspring going to have? Well, they're all capital G, lowercase g. The dominant allele is green. The dominant green peas are dominant to yellow peas. So 100% chance their offspring will have 100% chance of being the color green. Here's another example. When we're looking at probability, the Punnett square is showing you the probability of an offspring. So it'll show you the probability that the offspring will have a specific genotype. Being tall is dominant to being short. So remember, you use the letter for the dominant allele. So capital T equals tall, lowercase t equals short. So let's look at the parents. This parent, capital T, lowercase t. So they are heterozygous. Same thing with this parent, capital T, lowercase t. They are also heterozygous. So their genotype, both of their genotypes are big T, little t, and both of their phenotypes are tall, because tall is dominant to being short. So if we do this cross, and I'll do them in different colors, well, we have 3 different possibilities. So then we would look at the probability, the percentage, and the ratio. 1 offspring had TT, 2 offspring had big T, little t, and 1 offspring had little t, little t. So what is the genotype percentage? Well, again, each box is worth 25%. 25% chance that this offspring will have the genotype big T, little t, and will 25% chance that their offspring will be homozygous dominant. 2 boxes had big T, little t. 50% chance that their child will be heterozygous and have a big T, little t. 1 small t, again, that would be 25% chance that their child is homozygous recessive. So the ratio would be 1 to 2 to 1. So those are the percentages of the genotype. But what would be the percentages of the phenotype? Well, there's 2 possible phenotypes. The offspring can be tall or the offspring can be short. And what is going to tell us what the phenotype is? Well, if they have a dominant allele or not. Here, dominant, and here's a dominant. So 75% chance their offspring will demonstrate the phenotype of being tall, 25% chance that their offspring will be short. So that's why we determine the percentage of the different genotypes versus the percentages of the different phenotypes of what they will actually demonstrate, right? Because that goes up to this point up here, that because of the different effects of dominant and recessive alleles, an organism's traits do not always equal its genetic composition. We need to distinguish between an organism's phenotype, the physical appearance, versus the genetic makeup, the genotype. So for homework, I'm giving you a couple of practice problems with monohybrid Punnett squares before we move on to more difficult Punnett squares. So that's the purpose of going through all this, because it's going to get more complicated. There's going to be more factors involved. We're going over some more vocabulary to make sure that we have this down before we move on. And this is the first example from your homework, so we're doing it together, so that way it gives you an idea of what you're doing for the other problems. In silkworms, a single gene determines the color of the cocoon. The yellow cocoon allele is dominant, so it would be a capital C. Usually it would be the color, it would be a capital Y. I'm going to change that in your homework, but it's the same, it'll be the same problem. Indicated by a capital C for now until Miss Carrie changes that on your homework. Capital C equals yellow because it's dominant. The white allele is recessive, and it would be indicated by a lowercase c. Complete the following Punnett square, assuming that a homozygous dominant male is mating with a homozygous recessive female. Okay, first thing you're going to do, even if it didn't tell you the letters, homozygous dominant. 2 dominant alleles, capital C, capital C. Homozygous recessive female, 2 recessive alleles, lowercase c, lowercase c. I'll do it in 2 different colors. Fill in the Punnett square. So see how I put the 2 alleles from mom on one side, the 2 alleles from dad on the other. So we have a capital C, lowercase c in this box, capital C, lowercase c in this box, capital C, lowercase c in this box, capital C, lowercase c in this box. So what genotypes do we have? Well, this one's easy. We only have one genotype, big C, little c. What phenotypes are we going to have? Well, we only have one. Big C is there, so the dominant trait is going to be the phenotype, so it would represent the yellow phenotype. The color of the cocoon would be yellow. So what's the percentage of the genotype? They will be 100% big C, little c, which is in genetics terms heterozygous, and the phenotype would be 100% yellow. Okay, so work your way through the problems. Let me know if you have any questions. I want to dedicate today to review. Okay, so practice perfecting these skills before we move on.